Hello, everyone, and talk session. So, our webinars, as you all know, are part of now Interpol Young Academy and also aligned with Geoscience and Geoenergy webinar series run by Hardy and Sebastian. So, my name is, as you can see from my tag, Kamalijt Singh, and today in our studio we got Arjen Mascini from Ghent University and Mohammad Nooraiepour from University of Oslo. So they'll host today's speaker with me in the studio. So, so I'll move on to the first speaker. I introduce our first speaker is Ioannis Zarikos. Uh, he is a postdoc at NCSR Demokritos. He received his BSc in Environmental Earth and Environmental Sciences from National Kapodistrian. I hope I'm saying correctly. University of Athens,
Correct.
Greece. Okay, uh, and he did his joint MSc in Earth Sciences from Ghent University and Catholic University in Leuven in Belgium. So he then moved to join the environmental hydrology group, Utrecht University in the Netherlands. Actually, he moved around many different labs, which is quite nice to see, to continue his work in on multi-phase flow in porous media. And he received his PhD in 2018. So he currently works in microfluidic manufacturing and also multi-phase flow, both in natural and artificial porous media. So he's going to talk today on very interesting topic on about reservoir-on-a-chip. So welcome, Ioannis, and really thankful to you for accepting our invitation. We are looking
Thank
forward
you.
to see your presentation. So floor is
Okay.
yours, please. Mhm.
Thank you. So, as you can see from the title, we are going to talk about how we create a 3D printed Pore-like micro model using 3D printing technology. Now, I'll give you an overview of the presentation. First, we're going to talk about a brief review on the microfluidics and how they developed over time. Then we're going to focus mainly on the 3D printing techniques and which can be used for microfluidic production. We'll continue discussing how we're going to generate a pore structure that can be used for later for printing the macro model. Then we'll continue with the characteristics of the produced macro models. And then we're going to compare the two main techniques that we used, how they looked under micro-CT, and if we came into some problems with it. The two-phase flow test that we did on them, and of course how they performed. Now, microfluidics models actually. Then we have models which require the CNC machine to be generated. And then we move to optical lithography, etching methods, and soft lithography until we come to stereolithography, or as we know it, 3D printing. Now, 3D printing has many techniques. The most common one that everybody knows is FDM, fused deposition modeling. It's actually the cheapest and most simple way to 3D print these days. But in our case, there are only two techniques that can be used for microfluidics: is stereolithography and material jetting. Now, these techniques have some advantages over the rest of the techniques. First, they can create watertight structures. So in our case, we want the fluid to stay in the pore space. And they both use UV light source and photopolymer resins. The main difference between the two techniques is that MJ printers, they only spray tiny droplets and they cure by UV lamp. While for the stereolithography technique, we have a bath of photopolymer, and then with a laser, we point cure the area that we want it to be hardened. So it's slightly different. Now, SLA printers have commercial SLA printers. They can go down to 9 micrometers, while for MJ printers can only go down to 25 micrometers resolution. And that's a resolution up only on the X and Y axis, not on the vertical direction. Also, MJ printers, also known as multi-jet printers, they use a support material like a wax that has to be removed afterwards. Now, I come back to the printers. Well, we need a pore structure to act as a porous medium. So to do so, we create a cubic domain. In this case, it was 600 pixels in all directions. And then we generated a pore distribution, which is analog to a sandstone. We only used two parameters to control the pore structure. It was porosity and correlation length. And after we created this pore space, we subtracted a core from the center of the cube of 200 pixels in diameter to create our pore space. Now, the pore space we used, well, the micromodel had a porosity of 45, a permeability of 6.46 cubic square meters, an average grain diameter of 2.6 millimeters, and a correlation length of 35 pixels. Now, I understand that for most of us, a grain size of 2.6 millimeters is relatively large, but it's sandstone scale. So we had to create a relatively large model of 4.5 centimeters in length and 1.5 centimeters in diameter. Now, as I said before, we used the two different techniques, material jetting and stereolithography. We used two commercial printers in this case. For multi-jet, as they call it from the company printing, we use a Project MJ P3600. Now we managed to print at a resolution of 25 microns in X and Y direction for 26 hours, followed by quite a lengthy process of removing the wax. In the case of stereolithography, we use the Formlabs Form 3. Again, it's a commercial printer, which allows 25 microns resolution in all three directions. The printing time was relatively faster, but we had a little bit of post-processing because we had to additionally expose the sample to UV to harden the external parts. And then we had to immerse in isopropanol in an ultrasonic bath to remove the residual photoresist or from inside the micromodel. And because we don't use a supporting material in this case, we printed the micromodel both parallel and perpendicular to the flow direction in order to see if there are be changes in the hydraulic properties of the medium. Now, the problem with material jetting is that we cannot use, as you can see in the picture below, a core holder integrated to the porous medium, because otherwise we cannot remove the wax. While in the case of the stereolithography, it was very easy to have a solid, let's say, plug-and-play microfluidic device. Now, these are the results. On the left, you can see the material from the material jetting technique. It looks like a sandstone. It also has a light color, but because of the wax that we use, you can see that at some part of the model to build the microfluidic device with a core holder and some lure locks on the sides, so a complete way of a microfluidic device. Now, we had, in collaboration with Stuttgart, University, we scanned the microfluidics device under micro-CT. And first we scanned the multi-jet microfluidic device, but we weren't very happy with the pore structure, as you can see in the pictures. Now, we're still not sure what the problem is. We speculate that it's the process of removing the wax that probably cracked some of the pores. But currently we are working with different resins to see if we can solve this problem, because despite the lower resolution and the wax issue, it's still a reliable technique to make microfluidic devices. Now we use the stereolithography printer for the second part of our attempt to make a 3D printed microfluidic device. As you can see, we don't have any cracks here. Of course, it's a different material that we used in this case. It's high-end, very robust resin, usually used for plastic moving parts, so we didn't expect to have any issues. And as you can see, the pore space is quite nice. And, well, we believe that this is the go-to method to make a device that can be used for multi-phase flow experiments. Now, we then decided to do a flow experiment. So most of us are familiar with the CD scanner, but I will just describe a little bit. So we used the core holder, which in this case is integrated to the microfluidic device on a rotating stage. We added 3 wave valves on the top and bottom to allow fluid flow. And then we had an X-ray source and an X-ray detector that used to visualize a sample. And as you can see here, we have a quite nice result, as expected, of a water-wet microfluidic device. We only use water and air at this stage, which is current right now. And we are actually happy to analyze the rest of the data in the coming weeks. So to conclude, 3D printing can be used effectively to create a true 3D Pore-like micromodel. And I say true because we know that there have been attempts to make 3D printed microfluidic device, which are actually very close to the 2.5D or quasi-D microfluidics that we already use, probably made out of PDMS. The resolution that producing microfluidic device, and I think we have enough pores to represent a natural porous medium. So, like I said, stereolithography is a go-to technique to make these micromodels. And also, since it's an evolving technique, we have a variety of photopolymer resins that can be used, either softer or harder, or some of them with different surface properties. So we'll continue making microfluidics out of 3D printing and to tackle the different scientific questions we have to answer. Thank you for your attention. Questions?
Thank you, Ioannis, for this really great talk and introduction to probably the future of this field. Really looking forward to see more results. Unfortunately, there was a little bit of breaking of voice, but we managed with the internet connection.
Yeah. Sorry about that.
No, it's not your fault, I think. But we more or less, I think 99% was perfect, like with the internet connection.
Okay.
So we got a question to start with from Majid. He says he thinks that large sandstone grains of about millimeter size have micro to nano porosity. Can you comment on the potential effect on any conclusions you may have from these micro models?
Well, the thing is that we wanted enough pores to make a microfluidic device that is representative, right? Because if you can, usually we use sandstone samples of 6 millimeters in diameter. I think Arjen can comment on that. But for us to have enough grains, you use these larger samples. Now, for now, we try to replicate a simple sandstone. So nanoporosity, in this case, we don't have it because if we used the traditional technique when we melt plastic, we could have actually microporosity and nanoporosity because of the different spotting. But I don't think we can do it right now.
No, that's okay. Thank you. And I think in the future, maybe it may represent then carbonates, which are complex, have intragranular porosity sort of stuff. I think second question is, I think you answered, I missed the size. I think you mentioned about the size of the sample.
There are other 3D
So your connection was breaking. So I can repeat. I can just
very
mention the.
large sample.
Um, Jonas, I think his connection is breaking up quite a bit now. Okay, we'll wait for 30 seconds. Otherwise, we can move on. So I think for Majid's question, Arjen, in fact, do you have an answer? Because I think you use because no, he's back. Okay. Sorry,
Okay.
Jonas, we missed quite everything you said about the size of sample. The
Yeah.
question actually came from
Okay.
Majid.
Yes. So
So what he was asking, how
yes.
large can sample be, and is it still suitable for imaging?
Yes. So I think it has to down to the city that we are going to use. Actually, we can go larger. Usually that's a limitation of the printer. So in this case, it could be 20 centimeters tall. It would be okay, but then it has to do with the city if we can actually scan it nicely at a relatively high resolution. We can reduce resolution to scan a large sample, but small pores, we're not going to be able to visualize them. So it has to be a combination of both CT scanner and 3D printer. So we can go large, but then it's up to the CT scan to visualize it.
Okay. Thank you for this. I know I've got one question, or no, two questions. Well, I'll
Yes.
ask first one and then ask other studio people if they have any questions. The first thing, it's nice that you showed that you tried both the direction for printing,
Yes.
perpendicular and the same direction. I was wondering if you repeat those samples printing, do you get the same structure or do you see some differences?
We did once, to be honest. So we did one perpendicular, one parallel. When we measured permeability, it was is almost the same. It was very small difference, and we didn't do it again because we wanted to make sure that we can actually use it um, in the city. And of course, as you can see, we don't have too many pores. So we're doing another attempt to create a much finer pore space to have more pores. And once we reach but an representative volume of a sample, then we can multiply it. Replicate the process to see if we have consistency over the different over all the micromodels. But we expect to have the same pore structure every time we print the same STL file.
Okay. Thank you for that. I think resolution is quite nice. It's about 25 to 30 micrometer, which is quite good actually.
And it's relatively an economic solution. I mean,
I think okay, we missed the last part. Jonas, you said. Uh, can you hear us?
yes, I can hear you. Yes.
Okay, let's move on to the next question from Ishan Marker. So, did you see any impact of the type of resin you use on the wettability of the media? Like in general, was it water wet, or depending on different material, you expect different wettability?
Um, we use only one material, so this gray resin, and this one is water wet. But this one that it came with the printer. There are some other resins that I know they're selling with the printer that they are hydrophobic for the. But of course, it depends on the fluids that we are using, right, in the experiment. But in this case, we wanted a water wet, sorry, sample.
Okay. Before I make it open to the studio, I've just because it's along the same line, I have a question. Your material, let's say you put into your core holder, and then you apply your confining stresses. Do you think it will deform to some extent?
No, actually, I think I checked it last time. It was, well, this is very strong. There are some resins that are actually softer that can be deformed, but this one can go, I don't remember the value correctly, around 2.2 gigapascal tensile strength. So it's pretty strong.
Okay.
But there are resins that are made for molds that can be used, let's say, for making deformable porous media, and they can deform either elastically or plastically, so you can actually have the material to rebound to its previous state.
Okay, thank you very much. Uh, Mohammad and Arjen, do you have any questions or any comments? If not,
Yeah,
then oh yeah, okay,
I
Arjen
got
has.
maybe one small question. Is it possible maybe to use different polymers on the same sample and thereby sort of creating a mixed wettability or like different
So for
properties?
the, I think that for the SLA printer, there is a way to use two resins actually, but I'm not 100% sure. And for the multi-jet, we only used, you need the wax as support. I don't know if there's a chance to use two different resins to, for example, instead of change the settings, but I'm not 100% sure.
Okay.
Because in the SLA you have a bath with all the resin inside, and the stage is going upwards. So if you have them mixed, it will be an issue. Actually, I don't know. It's complicated.
Yeah, but it would be really cool if it would be possible.
I
Okay.
think that would be very nice, interesting idea. Yeah. Okay. Thank you, Ioannis. We've covered all the question, and it's also time to move on. Just wanted to highlight there's some appreciation from audience, from Andreas saying nice work, and also Majid is saying thank you and great job. With that, I would like to thank you for giving this great presentation and also taking some time off to discuss about your work. It's really interesting, and in the future, I would look forward to seeing more of the work, and maybe we can use it in our micro series. Yeah. Thank you. Okay, so we'll thank you very much once again. We'll move on to our next speaker. We got our second speaker, David uh, Scheidweiler. I'm so sorry if I pronounced that
Correct.
wrong. That's fantastic. Okay, so David has a background in chemistry from University of Bologna. But then he moved to explore ecology and did his PhD in this field from EPFL. So in his PhD, he investigated the morphogenesis of biofilms in sedimentary environment, which is a really interesting topic. So currently, he's a postdoc at the University of Lausanne in Switzerland. So where he's studying how microscale physical constraints impact microorganisms in porous environments. He's using for these things we're using microfluidics and microscopy. So today he's gonna present or give presentation on uh, microscale biophysical drivers or microbial colonization in porous media. So, welcome, David, and we're really thankful to you for accepting our invitation, and we look forward to your talk. The floor is yours, or the screen is yours.
Thank you very much for the inviting me and for the introduction. So, as you just said, the topic of my presentation today will focus on how the physical structure of porous system can drive microbial transport and so the colonization and the growth within these environments. So I think we all know that microorganisms are very important. They perform like a multitude of functions necessary to sustain the life, like they control carbon and nutrient cycling of aquatic and terrestrial environments. They interact with the living organisms they inhabit, like on the plant roots or even in our guts. The study of microorganisms relied extensively on a well-mixed system, which is a very powerful tool to describe process at a macroscopic scale. However, like soils, sediments, and biological tissues are all characterized by like a spatial heterogeneity where the physical constraints as flow or resources availability are heterogeneous and can span over several order of magnitudes across few micrometers, which is actually the scale that microorganisms experience. So I will start by showing you how microorganisms characterized by different dispersal traits, which I will now explain what it means, respond to flow heterogeneity typical of many porous environments. So we design a microfluidic porous channel where the configuration of the pore throat promotes a disordered flow, as you can see here by the normalized flow velocity in the log scale. We then injected a mix of motile and non-motile Pseudomonas putida cells suspended in a motility buffer and tracked the arrival time at the channel outlet. So by plotting on, you can see the graph on the right, the normalized concentration over time, we can see that macroscopically, motile cells traveled on average faster and got less retained than the non-motile counterpart. So this was an agreement with the pore scale observations, which show how motile cells exhibited a higher speed comparing the velocity distribution extracted from the trajectories. But motile cells, however, did not only swim faster along the system, they also explored broader space by swimming, as confirmed by analyzing the tortuosity of this trajectory. So overall, this enabled them to actively explore the space for resources or also evade regions with unfavorable conditions. In this disordered flow, bacterial cells exhibited a rotation while being transported. So while the non-motile one passively rotated by means of the flow shear they experience, interestingly, motile cells were, they reoriented themselves along the flow trajectory. And by analyzing the angle between the trajectory and the cell body, we saw that motile cells were more likely to be aligned towards the flow direction, which is the 0-degree angle in the two plots. So this means that on average, more active swimmers were aligned along the flow direction, thus explaining the faster travel times observed macroscopically. Finally, we linked the pore descriptors with macroscopic scale data with a stochastic model. Very briefly, so this mathematical framework, within this mathematical framework, the travel time of each bacteria is computed from 3 parameters randomly chosen from the distribution. And these parameters are the velocity that the cell is experiencing while being transported, the length at which the cell has a constant velocity, and the attachment rate. So the experiment that I showed you until now were performed in the time scale of hours. But if time is long enough, and once the dispersed bacteria, let's say they find a favorable environment, they may grow into colonies and behave very differently than single cells do. So in this different experiment, we use within a millifluidic porous channel, we injected a suspension of stream microbial cells, and we studied their growth over 10 days. So the biofilm initially coated the grain surfaces into what we call base biofilm, which is the red architecture you can see in the animation. And then from there, the biomass was extruded by the flow into these elongated architectures known as streamers. So overall, increasing the space exploitation for the entire community. Interestingly, however, we saw that these two architectures follow different dynamics. So the base biofilm could be described by a population growth model, which basically explains the binary division of microbial cells, while the streamer dynamics followed a linear increase that we described as a physical filtration model. So how it works, as the flow extrudes the biofilm from the grain surface, these formed filaments will cross the flow and increase in size by filtering the cells flowing by. We notice, however, another mechanisms which allow the biofilm to gain biomass. Once the grain surface was fully colonized, the base biofilm increased its surface, forming this kind of crested structures. So we measured this, what we call tortuosity of the perimeter of the base biofilm, defined as the ratio between the actual base biofilm perimeter and the grain perimeter. And we could see in the plot that the higher was the final tortuosity of the base biofilm, the higher was the biomass achieved. So this suggests that these crested architectures promoted biofilm growth by enhancing the surface exposed to the mass transfer. So in order to study the impact of resources heterogeneity on microbial transport and growth, I recently started to work with pore geometry with spatial heterogeneity that now is given by the grain morphology rather than by the pore throat sizes, as I showed you before. So in this system, two main pore classes are present. Pores where the flow is mainly advective. We call them transmitting pores, here highlighted in green, and dead-end pores where the flow is stagnant, as you can see from the velocity map. However, in the dead-end pores where we thought that transport would be mainly diffusive dominated, we observed actually vortices of secondary flow that control the transport time scale of polystyrene microsphere through the pore system. So, but we then wanted to study microbial transport through this geometry, not simply microsphere, polystyrene microsphere. So we saturated first this system with a motility buffer, and then this motility buffer allowed the cell to swim, but not to grow, just to explain, and then injected a mix of motile and non-motile Escherichia coli cells suspended in the same buffer. So over 2 hours of experiment, we recorded the microbial abundance through the system, and we saw that the motile population got retained around 4 times more than the non-motile cells. We basically use the non-motile cells as a passive tracer control. So basically, the higher diffusivity of the motile one allowed them to explore also the dead-end pores, and so increasing their accumulation through the entire system. So we know that flow heterogeneity can sustain chemical gradients that often can be also sensed by bacteria. So in the left panel, we see like a concentration map of a tracer being washed out by another solution in this channel. And you can see the strong gradients of the tracer occurring in time in proximity of the dead-end pores, where of course the residence times is much higher. So in this experiment, we injected a mix of motile and non-motile cells suspended in a motility buffer, but displacing a resident solution of an amino acid, it's serine, the amino acid, which is a chemoattractant for E. coli. So E. coli is attracted by the concentration, by gradients of concentration of this amino acid. So we recorded the temporal displacement that you can see here in the 2 animation of the bacteria across the 2 pore classes and quantified it. So the plot on the right now shows the ratio between the concentration of the bacteria in the dead-end pores versus the concentration of the transmitting pore. So we can see that the presence of the chemoattractant did not affect at all the distribution of the non-motile control, the 2 curves here on the bottom. Differently, the presence of the chemoattractants sensibly increased the abundance of the motile cells in the dead-end pore. So overall, we see how like disordered flow that can translate into resources heterogeneity in pore system may have profound impact on microbial, temporal, and spatial distribution and dynamics. So concluding, we have seen in section 1 that microorganisms with different dispersal traits, so the motile and non-motile, responded very differently to the flow heterogeneity. In the second section, I try to show you how the combination of biological and physical mechanisms are at the base of this space exploitation of microorganisms in microbial biofilms in porous systems. And finally, dead-end pores, which are, let's say, generally considered as stagnant regions, actually they present transport properties which can be very relevant for microbial activity. So finally, I would like to take the opportunity to, of course, thank the people involved in these works, my colleagues of the laboratory of Professor Pietro De Anna at the University of Lausanne, as well my former supervisors at EPFL. And for those interested, I leave here also my email and a link to my website where you can find more information. With this, I thank you, and I'd love to take your questions.
Thank you, David, for this wonderful talk. It was really interesting and comprehensive, and it's in line of the wonderful work that you are doing in the Environmental Fluid Mechanics Laboratory at Lausanne. Very nice. First, we have the first question from Andreas Iotis. He's asking, what is the difference between motile and non-motile bacteria?
Yes, sorry, I was a little bit quick in explaining this. So basically, I always use the same bacterial strain, which, however, has been genetically modified not to produce the flagella. So I think we can know that bacteria swim by the use of certain flagellas, and like the non-motile one was like a flagella deficient organism. So basically, it's just a rod-shaped cell that passively is transported by the flow that it experiences.
I have one question of my own. Can you elaborate more on the processes and the phenomena that you saw in the dead-end pores? Are they limited to the bacterial environments, or when we are studying reactive transport in porous medium, or generally fluid flow and transport of solutes in the porous medium? Can we expect the same behavior in the dead-end pores?
So if I understood your question, you wanted to understand, for instance, the vortices that we have seen in the dead-end pores, or you're talking about the microbial transport mechanisms related to the chemoattractant?
Mostly the first one, and also about this secondary flow that you observed in your experiments.
So what we thought initially is that, like in the dead-end pore, the flow would be stagnant. So like particle transport, particle like being seeded in a dead-end pore would be released from the dead-end pores following simply diffusion mechanism, so a diffusion time scale. But actually, we have seen that unfortunately now I haven't prepared a slide for that, but maybe I have something. Wait a second. Here I have it. So from experiments and simulation, we have seen that like the particles, now these are microspheres of polystyrene, like the tiling of those particles is the tiling response, are the particles responsible for the time scale of the tiling of these breakthrough curves. So this is the normalized escaping time of particles from the system at the outlet. And so we with simulation and with an analytical model, we have been able to show that actually these secondary vortices are the one responsible for this late arrival time. Because if it would be diffusion only, it would be like a faster process.
Wonderful, perfect. Now I open the questions from the host, from the studio. If you have any question, Kamal or Arjen, please go ahead.
I had one, but you stole my question. I think it's quite clear now, but it's very interesting as well. So you see these vortices, even the slow velocities are there. So they can impact.
Yeah, we were very surprised.
Yeah, that's very interesting. We found something very similar, but we are still exploring, but not related to bacteria or microorganisms. Second thing is, it's not a question. Maybe I just wanted. So what my understanding now here is that your flow is going. You got the micro channel, or let's say microfluidic device. You got plates on the top, bottom, and you're creating the biofilms around the pillars. right?
Yes.
So that means you need an obstacle to form the biofilms. Otherwise, you have surfaces on the both and the top and bottom, but you don't see any biofilms forming there, right? So roughness doesn't have anything to do with that, probably, or maybe it has to do because maybe your plates are very smooth. So it's, do I understand correctly? You need obstacles in the flow path so that somehow you can nucleate those biofilms.
Yes. So maybe it was not very clear in the presentation. So exactly, the all the microfluidics or millifluidic in this case that experiments that they have been performing use like a pore system where like these pillars are the obstacles towards flow.
Yeah.
And this is like basically simulating somehow like a 2D cross section of a porous environment. So we observe a system which we can resume as being a 2-dimensional, but of course like is a 3-dimensional system because like for instance in this case like the height of the system is 400 micrometers, 0.4 millimeter. And so we basically reduce the heterogeneity of the flow to a 2D system because like the main flow diversity is across the flow path that has to evade the little grains.
It's okay. Thank you. I think I might have missed initially, but sorry about that. But but thank you. Thank you once again for explaining this. Yeah.
Okay, I think now it's time to wrap up the session. Thank you, David, for the wonderful presentation again, and
Thanks to
for the
you.
discussion afterwards. Now we can move on to the speakers for our next session on November 16th. We have two interesting talks by Federico Lanza on non-Newtonian rheology in a capillary tube with varying radius from Université Paris-Scaglier, and also PoreLab NTNU in Norway. The second presenter is Lucas Moser. He is going to talk about deep generative models for stochastic image reconstruction and inverse problems. He is now in Earth Science Analytics, and he was formerly a PhD student at Imperial College London. With that, I would like to thank you for being with us today. This is the Porous Media Tea Time Talks team, and as you can see, we are part of the Interpore Young Academy. Thank you again, and hope to see you in our next session on November 16th. Have a nice day. Bye-bye.
Bye.